Hey guys, today we are gonna kind of give you an introduction to metabolism. So to start, remember that metabolism refers to all of the chemical reactions that occur in an organism, and it's what we would consider an emergent property because obviously metabolism is this complex thing with a lot of different kinds of chemical reactions, and no single chemical reaction can create metabolism, but they all contribute to it. So in living systems, we talk about metabolic pathways because it's usually, to get to an end product, it usually requires multiple steps. So this is kind of your generic model of a metabolic pathway. You have some reactant or starting molecule, and then there's an enzyme that will perform the first, chemical reaction on it, or I should say catalyze the first chemical reaction on it, and that creates a product B, which is different than A, right? And then there's a second enzyme that can come in and facilitate the second chemical reaction, and then you get product C and so forth. And so these metabolic pathways will, they can be, in this case, we've got like 3 steps. They could be 12 steps. They can get really complicated. But in the end, you reach the final product, which is the thing that the organism actually needs. And these can be regulated at each of those steps, and we'll look at that more a little bit later. So if you remember, we talked about the difference between catabolism and anabolism. Remember that catabolic pathways are digestive. They break molecules down into simpler compounds, and in doing so, they have a net release of energy that can be used to do work. So for example, cellular respiration is a really important catabolic pathway that breaks down glucose in the presence of oxygen to generate ATP, which of course we use to provide energy for lots of our metabolic chemical reactions. And then our anabolic pathways require energy, right? You're making a higher energy product, building a more complex molecule from a simpler one. For example, synthesizing proteins from amino acids, or copying your DNA, things like that. And so in living systems, what we have to do is we have to kind of link a catabolic pathway to an anabolic pathway, and that way we can provide the energy to create the complexity of life. So just a quick note about energy. Energy exists in lots of forms, right? Kinetic energy is associated with motion. Potential energy is the energy that matter possesses because of its location or structure. Of course, there's heat or thermal energy, and then chemical energy is the potential energy available for release in a chemical reaction. And we can convert energy from one form to another, and that's really important for life, right? If you think about photosynthesis, you convert light energy into chemical energy, essentially, and then we can use that chemical energy to generate kinetic energy, right? So that we can move or whatever, or to generate thermal energy to maintain our body temperature or whatever. Okay, so this just kind of shows you the difference between potential and kinetic energy, right? A diver is going to have more potential energy at the top of a diving board than somebody standing, on this deck, or somebody who's already jumped into the pool. When the diver jumps off of the board, the potential energy is converted into kinetic energy, energy of motion. Okay, so thermodynamics is the study of energy transformations. And if you took chemistry, you probably learned a little bit about thermodynamics. And of course, life has to obey the laws of both physics and chemistry. So according to the first law of thermodynamics, the energy of the universe is constant. So energy can be transferred and transformed, but it cannot be created or destroyed. So the first law is also called the principle of the conservation of energy, right? And so that just shows you, again, we can convert energy from potential to kinetic to chemical to thermal, right? We can convert between types of energy, but we're not going to create new energy or lose energy. So during every energy transfer or transformation, some energy becomes unusable and it's often lost in the form of heat. So the second law of thermodynamics says that every energy transfer or transformation increases the entropy or disorder of the universe. This just kind of shows you, right? So chemical energy is taken in, right? The bear eats the fish, and through the process of cellular respiration, we can generate energy in the form of ATP, but a lot of that energy is lost as heat. I don't know if you remember baby bio, we kind of teach you in a food chain that only about 10% of the energy we eat is usable, and the other, 90% is going to be lost in the form of heat. So living cells unavoidably convert organized forms of energy into heat. Now, spontaneous processes are going to occur without an energy input, and they can happen quickly or slowly, but they don't need an energy input really to happen. And for a process to occur without an energy input, it has to increase the entropy of the universe. So biologists want to know which reactions will occur spontaneously and which require an input of energy. So to do so, they have to determine the energy changes that occur in chemical reactions. So a living system's free energy is the energy that can do work when the temperature and pressure are uniform. And free energy change is measured as delta G. So maybe you guys know this from math or something, but the little triangle sign here, delta means change in, right? And G is going to be our free energy. So delta G means change in free energy. And the change in free energy during a process is going to be related to the change in enthalpy, or the change in total energy. Change in entropy is delta S, and temperature in Kelvin is T. And so here is your equation for change in free energy. Delta H, which is your total energy, minus temperature times delta S, or entropy. So here's the thing. I have not seen an AP biology test ask a student to calculate this since I started teaching AP biology. It is on the equations sheet, so it's something you want to be familiar with, but I won't ask you to calculate this on a test personally. Here's what you should know though. You should know that if a process has a negative delta G, it's going to be spontaneous. It'll happen without a net input of energy. So spontaneous processes are the processes that can be harnessed to do work. So those spontaneous processes tend to be called exergonic reactions. An exergonic reaction proceeds with a net release of free energy. So I always think EX, like exit. Energy is going to exit the reaction. Endergonic reactions are the opposite. They require an input of free energy in order for them to occur. So these are non-spontaneous reactions. All right, so again, going back here, spontaneous processes are going to have a negative delta G. So your exergonic reactions are going to have a negative delta G. Your endergonic reactions are going to have a positive delta G. All right, and so if we kind of look at what this could look like graphically, because of course graphs are a great way to model, energy changes over the course of a reaction, you can see that an exergonic reaction has a higher energy reactant and a lower energy product. And so the amount of energy difference between the reactants and the products, that's going to be your delta G. Endergonic reactions are the opposite. The products have more energy than the reactants, and so that energy had to come from somewhere, and so that is why they're endergonic. They require energy to enter the reaction, right? And so again, the change in energy between the reactants and products is going to be your delta G. So reactions in closed systems eventually reach equilibrium, and then they do no work. So cells are never in equilibrium. They're open systems. They experience a constant flow of materials, and a defining feature of life is that metabolism is never at equilibrium, right? We use our metabolism to keep ourselves from falling into equilibrium. So in a catabolic pathway in a cell, you're going to release free energy in a series of reactions. And closed or open hydroelectric systems can serve as analogies for this. So a cell will do three main kinds of work: chemical, transport, and mechanical. And in order to do work, we have to couple exergonic reactions, which release energy, with the endergonic reactions that require the energy. So metabolism is largely about coupling these chemical reactions to maintain homeostasis. So most energy coupling in cells is mediated by the molecule adenosine triphosphate, ATP. So if you remember, phosphate groups have a negative charge. Also remember that ATP is fundamentally a nucleotide. So here's your sugar, here's your nitrogen base, and then here are your three phosphate groups. And because they're negatively charged, but they're also covalently linked, remember that opposite charges attract, but when you have the same charge, they repel each other. And so holding those negatively charged phosphate groups together with these covalent bonds creates a lot of potential energy in there, right? Chemical energy is the potential energy of the molecule based on its structure and properties. And so there's a lot of energy stored there. And these phosphate groups would rather not be bonded to each other. They would rather be bonded to something else, right? And so we can harness the energy in ATP by hydrolyzing the bonds between the phosphate groups. So hydrolyzing, remember hydrolysis is when we use water to split a bond, and so water comes in, and here you could see we've hydrolyzed one of these phosphate groups off. We are going to call it inorganic phosphate now, and that's going to lead to a net release of energy, and that energy can be coupled to an endergonic reaction to drive it. So the bonds between the phosphate groups of ATP's tails can be broken by hydrolysis, and energy is released from ATP when the terminal phosphate bond is hydrolyzed. In other words, it requires energy to break bonds, but when the phosphate group forms a bond with something else, the energy is released. Okay, so that's a fundamental principle in chemistry. Whenever you want to break a bond, energy goes in. Whenever you make a new bond, energy is released. And what we've done is, if we go back to this graph, right, when we hydrolyze ATP, ATP is your reactant, lots of energy stored in those phosphate groups that are linked together, right? And by hydrolyzing that terminal phosphate group, right, we create a lower energy product when the new bond is formed, and then that gives you the net release of energy, which can be used to do work in the cell. Okay. So this release of energy comes from the chemical change to a state of lower free energy, not necessarily from the phosphate bonds themselves, right? We create a product that has less energy than the reactant. Okay, so in the cell, the energy from the exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction. Overall, the energy-coupled reactions are exergonic. ATP drives endergonic reactions by phosphorylation, so you transfer that phosphate group to some other molecule, such as a reactant, and the recipient molecule is now going to be called a phosphorylated intermediate. In baby bio, I tell my students to think of phosphate group as batteries, right? And whatever it connects to or plugs into is going to receive some amount of energy. Okay, so how does this work, right? We can use ATP, we can transfer that phosphate group to like a transport protein, and that can allow it to pump molecules across the membrane, right? We also can use it to power motor proteins. Remember, we watched those motor proteins walking around along the microtubules of the cytoskeleton, right? And so we could do transport work across cell membranes. We could do mechanical work. You could think about the mechanical work used to contract your muscles, right? And so there's a lot of things that we can do with this ATP. And it's a reversible process. So energy from catabolism, some kind of exergonic energy releasing reaction, is used to convert adenosine diphosphate into adenosine triphosphate, so reactant product, right? And so a good example of a catabolic reaction that does this is, of course, cellular respiration, right? We digest glucose and we release a lot of energy through that catabolic chemical reaction, and we use that energy to generate ATP. And then ATP is our intermediate that we can couple to tons of different cellular processes that are endergonic, that require energy, right? And the transfer of the terminal phosphate group provides the energy to do the work, and then that converts your ATP back into your ADP and PI, inorganic phosphate again, and we can repeat the process using the energy from catabolism to generate more ATP. So enzymes speed up metabolic chemical reactions by lowering energy barriers. A catalyst is a chemical agent that speeds up a chemical reaction without being consumed by the reaction, and enzymes, of course, are catalytic proteins. And you already know this because we already did that kind of enzyme lab using the little foam pieces and stuff, right? Okay, so every chemical reaction between molecules involves breaking bonds and then forming new bonds. Your atoms basically are going to find new partners. The initial energy needed to start a chemical reaction is called the free energy of activation, also known as activation energy, or E sub A. So when you want to break the bonds in your reactants, you have to put energy in, right? And we have said this many times, always. Breaking bonds takes energy. So now we're giving that energy a name. It's called activation energy, and it's often supplied in the form of thermal energy or heat. So enzymes, what they do, the way they work, is they lower the amount of energy you have to put in to break a bond. They do not affect the delta G, right, the change in free energy. The energy in your reactants and the energy in your products does not change. The thing that changes is how much work you have to do to break the bonds in the reactants so that you can create the new bonds to form the product. All right, so you guys have seen this already, right? Energy in reactants, energy in products. That does not change. That is your delta G. But what we're going to add to our graph now is the activation energy, right? The amount of energy I have to put in to break the bonds in the reactants so that the new bonds can form. So you can see that without an enzyme, that energy barrier, the activation energy without an enzyme, the black line, this hump is higher, right? And if I use an enzyme, I don't have to put as much enzyme in. So you can see the red line, it doesn't go up as high. And because you don't have to put as much energy in, you also will get to your product a little bit sooner. Sometimes you'll see the x-axis labeled progressive reaction. Sometimes you'll see it labeled time, right? And so it doesn't take as much time to get to your product because you don't have to do as much work to get that reaction to go. Okay. Again, this should all be review for you. The substrates are specific for the enzymes because remember, enzymes have an active site, which is complementary in shape to the substrate. The substrate will bind the active site, forming the enzyme substrate complex. And remember that there are two kind of models to talk about this. There's the lock and key model where your substrate fits exactly in the active site. And then there's the induced fit model, which says that the active site might open up. And remember I said it's kind of like it gives the substrate a hug, right? It opens up and then sort of wraps around it. And both of those models are pretty accurate ways to think about it. Every enzyme's a little bit different. So one will be more induced fit, one might have to open up a bit more, and the other might, have a substrate that just kind of fits right in. And so this shows you what the induced fit model might look like, right? So there's this pocket, the active site for the substrate, and here you can see the substrate gets in and then the enzyme wraps around it. And so there's not just a structure function thing, but you have to remember, again, go back to that modeling activity we did, there is a microenvironment in this pocket, right? Maybe it's hydrophobic, maybe it's acidic, maybe it's basic, whatever it is, it's creating the perfect microenvironment for the chemical reaction to take place. So in an enzymatic reaction, the substrate binds to the active site of the enzyme, and the active site lowers the activation energy barrier by doing a couple of things. Number one, it can orient substrates correctly, right? So the correct part of one reactant is directly next to the part of the other reactant it has to react with. Two, it can put strain on the substrate bonds, right? Like it can kind of force them into a position where maybe that bond is having a hard time being held. It can also provide a favorable microenvironment, and enzymes actually can temporarily covalently bond a substrate. That is a temporary thing, but it can form a covalent bond with the substrate, right? And then that can allow the substrate to react with, something else. It can stabilize the intermediate. Now, enzymes, like all proteins, have a primary, secondary, tertiary, and quaternary structure. And it's important to remember that the covalent bonds found at the primary structure are very strong, right? There are peptide bonds that hold those amino acids together. But the secondary structure, the alpha helix and the beta pleated sheet, those are held together by hydrogen bonds, which are pretty weak bonds. And when you get to the tertiary and quaternary structure, if there is a quaternary structure, while you might have a disulfide bridge as a covalent bond, that's the only covalent bond found there. Everything else that is holding your three-dimensional shape of your protein together are pretty weak interactions, ionic interactions, hydrogen bonds, things like that. And so they are subject to environmental factors that might interfere with those weak bonds. So a change in temperature, a change in salt concentration, or a change in pH can influence the three-dimensional shape of the enzyme, and of course, structure determines function. So if you lose your three-dimensional shape of your enzyme, it's very unlikely that it's going to be able to do its job to catalyze that chemical reaction. The other thing that can affect an enzyme's activity is substrate availability, right? And this makes sense. If there's not a lot of substrate available, the enzyme can't create product very quickly. Enzyme concentration also can affect how quickly you get product, right? You might have tons of substrate, but if you only have a little enzyme, you're going to quickly saturate your enzymes, and they can only catalyze that chemical reaction at a certain rate. So each enzyme is going to have an optimal temperature, pH, ionic concentration that they favor, where they work best. And so the optimal conditions are going to favor the most active shape of the enzyme, and obviously the most active shape means you'll get more product more quickly. And so you'll see this on graphs, right? And so here they're going to show you the rate of reaction on your y-axis, temperature or pH maybe on your x-axis, and you can see we've got two different enzymes. So if we look at an enzyme that works best in humans, you're probably going to see an enzyme that works best around body temperature, which is 37 degrees Celsius, or 98.6 or something Fahrenheit. If you're looking at a thermophilic bacteria, though, a bacteria that lives in like a hot springs, their enzymes are designed to work at a much hotter temperature. Designed is a bad word. I should have said, they have adapted to have enzymes that work in a hotter temperature, right? The process of natural selection has selected for enzymes that are heat tolerant. And so those enzymes are going to work better at the hotter temperatures because, of course, that is gajillions of years of evolution, right, have selected for that. pH, right? So So we tend to think of our pH in our body as pretty neutral, right? But remember that the pH varies throughout the body. So enzymes that work in our stomach are going to work at a lower pH in a more acidic environment. And that's actually a safety mechanism, right? Think about that. Those digestive enzymes in the stomach, or even the lysosome, remember the lysosome is acidic as well. If they escape that environment, they're going to turn off. We don't want to digest ourselves, right? And so that maintaining the low pH, the high hydrogen ion concentration allows those enzymes to work properly. Now, if you go somewhere else in the body, like your intestines, which actually has a slightly basic pH, we're going to find enzymes in there whose optimal is slightly basic, right? So trypsin works in the intestines, and it works at an optimal pH of 8. Trypsin would never work in a lysosome or the stomach. And the stomach enzymes would never work in the intestines, right? And so that allows us to kind of regulate where these enzymes have their activity within the body. And enzymes that work in the cytoplasm of a cell are going to work best in a pH that's more slightly neutral, right? So a little bit, you could shift this red down a little bit, right, for something that works in maybe cell or in your blood, which is more neutral. Now, sometimes enzymes need helpers. And if it is a non-protein helper, we call it a cofactor. So cofactors can be inorganic things like metals. Magnesium is an important cofactor. Some of you might take a magnesium supplement, and it helps a lot of our enzymes do their job. If there is an organic cofactor, organic meaning, think carbon, hydrogen, and oxygen, right? We call it a coenzyme. So a lot of coenzymes include things like vitamins, right? And this is why you need your, your vitamins. Fundamentally, why we take magnesium and vitamins and things like that is because our body uses them to, help our metabolism, to keep our enzymes functioning at their best. You might also remember from the enzyme lab that we can inhibit enzyme activity either competitively or non-competitively, right? Remember, competitive inhibitors bind to the active site and compete with the substrate for the active site. When the inhibitor is bound to the active site, the substrate can't get in and the chemical reaction can't happen. Non-competitive inhibitors, remember, they bind to a different part of the enzyme, and that changes fundamentally the shape of the enzyme, often making the active site either less effective or less available. Examples of common inhibitors would be toxins, poisons, pesticides, and antibiotics. So here, this shows you normal binding of a substrate to an active site. Here's competitive inhibition, right? You've got something blocking the active site, and then here's your non-competitive inhibitor. It binds somewhere else on the enzyme, but you could see that affects the shape of the active site. So we need to be able to regulate enzyme activity, which is the whole point of these different kinds of inhibitions, right? So if metabolic pathways weren't tightly regulated, we wouldn't be able to have a homeostasis or metabolism, right? Chemical reactions would just occur willy-nilly whether we needed them or not. And so cells can regulate their chemical activity by turning genes that encode enzymes on or off. And so you might make more or less of an enzyme, or you can regulate the activity of the enzyme. So make sure we have the enzyme present, but turn the enzyme off when we don't need it, either through competitive or non-competitive inhibition. So one type of regulation is called allosteric regulation. And this can either inhibit or stimulate an enzyme's activity. Okay, so I want to repeat that. It can kick an enzyme into high gear so that it's really doing a lot of chemical reactions, or it could turn an enzyme off. And so again, think of enzyme activity not as a light switch that's either on or off, but as a dimmer switch. And we can control where that dimmer switch is. We can control how on an enzyme is. So allosteric regulation occurs when a regulatory molecule binds to a protein at one site and then affects the protein's function at another site. This is similar to non-competitive inhibition, except that it also could be activation, right? So most allosterically regulated enzymes are made from polypeptide subunits, which means most of them have like a quaternary type structure. Each enzyme has an active and an inactive form, and the binding of the activator can stabilize the active form. The binding of an inhibitor will stabilize the inactive form. And so here you can see, here's an enzyme that is showing you quaternary structure. You have 1, 2, 3, 4 different subunits. Each subunit has an active site, which is the little pocket. But it also has a regulatory site, not in the active site, right? This is somewhere else. And so you could see if an activator binds right here, that stabilizes all 4 of these subunits in their active form, and they will catalyze many chemical reactions. On the other hand, you could have an inhibitor bind to that regulatory site, right? So this is allosteric regulation. It could be you can activate the enzymes or you could inactivate them, depending on if you're using an activator or an inhibitor, right? And this, you can see it causes a shape change in all 4 of these enzyme subunits, and so your active site becomes inaccessible or inactive. Okay, so some relevant FYIs. Allosteric regulators are attractive drug candidates for enzyme regulation because of their specificity. And in eukaryotic cells, some enzymes reside in specific organelles. So I already mentioned to you that there are enzymes that work in the lysosome because there's a low pH in the lysosome, high hydrogen ion concentration. And so those enzymes work in the lysosome, but they wouldn't work outside of the lysosome. There are also lots of enzymes that function in cellular respiration located in the mitochondria, and you're going to see that there is an acidic compartment in the mitochondria. And so again, we can allocate enzymes to different parts of cells, and those different parts might have different microenvironments that either increase or decrease enzyme activity. Okay, so that first pathway that I showed you at the beginning of the lecture, now we make it a little bit more complex, right? You still have an initial reactant. The substrate is in blue. We're going to call it threonine, okay? That's an amino acid. So threonine binds to enzyme 1. It happens to be called threonine deaminase. You don't need to memorize that. But that catalyzes a chemical reaction. So here's your first intermediate. Then you need another enzyme, another intermediate. So multi-step pathway, you get a product. The product is isoleucine. Now, we don't need an infinite amount of isoleucine. So what happens is isoleucine actually behaves as an allosteric regulator, and this leads to what we call feedback inhibition, right? So the allosteric regulator binds to the regulatory site, changing the active site of enzyme 1, turning it off. So when you make enough isoleucine, you can stop making isoleucine, so you're not wasting energy building something you don't need any more of, okay? This would be an example of negative feedback, right? We talked about that in Unit 1. And you also can see examples of positive feedback, right? And it's not depicted here, but where your product can go kick that into high gear.